Okay, let's unpack this. Have you ever wondered what's truly at the cutting edge of genetic discovery? What are the tools and techniques allowing researchers to unlock the deepest secrets of human health at, well, just an unprecedented scale? Today, we're embarking on a deep dive into the advanced genetic methodologies currently revolutionizing genome-wide association studies, or GWS. It's not just about what's being found, but how it's being found, and honestly, that's where the real excitement lies. Our mission for you, the listener, is to provide a shortcut, sort of, to being well informed on the bleeding edge of genomics. We want to spark those aha moments and make these really complex concepts clear and actionable, all without that feeling of information overload you often get wading through dense papers. We've gathered some incredible source material just packed with breakthroughs, and our expert guide is here to help us navigate this pretty intricate landscape.
It really is a fascinating time in genomic research. The advancements in methodology are proving to be just as impactful, sometimes even more so, than the discoveries themselves. The sheer scale and the sophistication we're seeing in these new approaches are fundamentally changing how we understand disease at its most intricate molecular level. It's a
Wow.
field that's perpetually pushing the boundaries of what's possible. It's really exciting.
Absolutely. So let's dive into the foundational power that's enabling so much of this. I'm genuinely fascinated by what makes modern GWAS so incredibly powerful today. Can you maybe walk us through the core drivers of that immense scale and why it's just so critical?
Yeah, absolutely. What's truly revolutionary now is how GOS is being conducted. At its core, GEOS involves rapidly scanning millions of these discrete DNA markers, tiny variations in our genetic code across the complete sets of DNA, or genomes, from a vast number of people. The goal really is to find genetic variations that are statistically associated with a particular disease or a specific trait.
Like a massive spot the difference puzzle, but with DNA.
Exactly. That's a great way to put it. Instead of pictures, we're comparing millions of genetic sequences to find those subtle differences that might predispose someone to, say, type 2 diabetes, or even influence something like their height. And this approach, it's been instrumental over the past decade or so in identifying thousands of genetic regions or loci linked to common diseases.
Right.
We're talking about everything from neurodegenerative disorders like Alzheimer's and Parkinson's to cardiometabolic conditions, psychiatric disorders, even fundamental physical traits like height and body mass index, BMI. But the key, as you pointed out, is the scale, the sheer scale at which we can now perform these studies.
And when we say scale, we're really talking about these massive biobanks, aren't we? It's not just collecting more DNA, but collecting the right kind of data alongside it.
Precisely. Biobanks like the UK Biobank, FinnGen in Finland, the All of Us research program in the US, and the Million Veteran program. These are truly the backbone of this massive scale trend. These aren't just collections of DNA samples, they're incredibly rich resources. They link genetic information with extensive longitudinal health records, detailed lifestyle questionnaires, and just a wealth of phenotypic data, meaning observable characteristics and traits.
Wow, so millions of people sometimes.
Oh yeah, these resources often comprise hundreds of thousands to even millions of participants. And each one has this deep trove of associated health information. And this immense scale is absolutely crucial because many genetic associations with common diseases are individually quite subtle. They might only explain a tiny fraction of the overall risk.
So you need huge numbers to even see the signal.
Exactly. To confidently detect these subtle signals, you need a very large number of participants. Smaller studies would simply miss them entirely. They wouldn't have the statistical power. If we connect this to the bigger picture, this kind of scale allows researchers to move beyond just single gene disorders, which are relatively rare and often have a strong, clear genetic cause, like cystic fibrosis. Instead, we can now robustly investigate complex polygenic conditions. These are influenced by the interplay of many genetic variants, each contributing a small effect alongside environmental factors.
Right, like heart disease or diabetes.
Precisely. It's a paradigm shift in how we approach disease etiology, moving from thinking about a singular cause to understanding this complex mosaic of influences.
That makes perfect sense. But okay, to handle that kind of data, millions of genomes, billions, maybe trillions of data points, you must need some serious computational firepower and very specialized software. What are some of the core tools at play in modern GWAS and what makes them so fundamental?
Indeed. Processing and analyzing these vast datasets requires a suite of highly specialized software tools. Think of it as a finely tuned orchestra of algorithms, really. While there are many, some of the foundational names you'll hear are PLINK, GCTA, Rigini, SAGE, and BoltLMM
Okay.
to give you a sense of their function. PLINK, for instance, is often considered the workhorse for basic GWAS data processing and quality control. It handles everything from data formatting to allele frequency calculations, the real basic stuff. Tools like Origine and BoltLMM, on the other hand, they're designed for more complex statistical modeling, particularly with these very large cohorts. They efficiently account for critical confounding factors like population structure.
You mean the genetic differences between populations?
Exactly, the genetic differences that naturally exist between distinct human populations. And also relatedness among individuals in the study. You have to account for families. This ensures that the genetic associations identified are robust, statistically sound, truly meaningful, and not just, statistical artifacts. They're built to handle the computational challenges of millions of individuals and millions of genetic variants simultaneously. It's a huge computational lift.
Before we dive deeper into the analytical wizardry, let's quickly touch on how we even get this incredible genetic detail from all those biobank samples. What are the workhorses of variant identification that feed these massive studies? How do we read the DNA in the first place?
All right, good question. The primary methods for capturing this genetic detail, the ones providing the raw material for GWAs, are whole exome sequencing, or WEES, and whole genome sequencing, WGS. These are high-throughput sequencing technologies. They are absolutely crucial for comprehensively identifying all sorts of genetic variants, everything from common single nucleotide polymorphisms, SNPs, to rarer variants, small insertions or deletions, even larger structural variations, all of which might have significant impacts on health or traits.
And how are these applied in a real-world setting like the UK Biobank, which you mentioned earlier? Because I imagine there are different strategies for different research questions, right? You don't always need the whole genome.
Absolutely. The UK Biobank dataset, for instance, represents a really sophisticated integration of different genetic data types. It combines data derived from genotype arrays, which measure common variants, which are then imputed, meaning statistically inferred to fill in gaps, with actual sequencing data from WES and WGS.
Okay, so multiple layers of genetic info.
Exactly. To give you some specifics, the WES data targeted the coding regions of approximately 18,893 genes. Now, this is a crucial distinction, as the exome represents only about 1% of the entire human genome.
Just the protein coding bits.
Right, but it contains the instructions for making proteins, so variants here are often more directly impactful on function, and they sequence this deeply. Over 95% of the bases in these targeted exome regions were sequenced to a depth greater than 25x. That means each position was read, on average, at least 25 times, ensuring very high accuracy for calling variants.
Okay, that's deep. And WGS.
WGS, on the other hand, captures virtually the entire genome, both coding and non-coding regions, with an average depth greater than 15x. The result is a significantly larger number of identified variants.
Yeah.
Around 599 million from WGS, compared to about 17 million from WES in the UK Biobank context.
Wow, huge difference.
It really highlights the comprehensive nature of whole genome data, which is essential when you're looking beyond protein coding genes to, say, regulatory regions, or searching for rare variants in less explored parts of the genome.
So beyond just finding new variants, what are some of the specific cutting-edge applications of WES and WGS that researchers are leveraging today? What else can you do with that data?
Oh, their applications extend far beyond initial discovery, diving deep into understanding disease mechanisms. For instance, take the analysis of clonal hematopoiesis, or CH.
Okay, what's that?
It's a condition where a person's blood cells acquire mutations over time that allow them to expand, creating clones. And this can potentially contribute to conditions like blood cancers or even cardiovascular disease. Using WES data, researchers could identify somatic variants. These are mutations that arise after conception, present only in specific cell lineages using a sophisticated bioinformatics tool called MUTEC2. These variants were then meticulously filtered against a catalog of 15 known validated CH driver genes.
Yeah.
This detailed approach allowed for the detection of these crucial somatic variants across diverse ancestral groups within both the MCPS and UK Biobank cohorts. This is vital for understanding how these acquired mutations initiate and propagate disease processes in specific cell populations. WES and WGS are also absolutely critical for identifying rare variants associated with disease risk. Think pathogenic loss of function, or PLOF, variants in genes linked to Mendelian cardiomyopathies.
These are severe heart conditions often caused by a single rare gene change.
Exactly. Highly impactful genetic variants. And WGS specifically was also used for looking at longitudinal telomere length, or LTL, in GWAS.
Telomeres, the caps on our chromosomes.
Right. Their length is an indicator of cellular aging and disease risk. So WGS helps investigate the genetic factors influencing telomere length. It just demonstrates the incredible versatility of this sequencing data in answering very different biological questions.
So with these massive data sets from WES and WGS, you might think, okay, we're all set for perfectly accurate findings. But even with incredible scale, inherent biases can creep in. I'm thinking about volunteer bias, which sounds like a significant challenge in these large studies. How do researchers tackle that kind of hidden distortion?
That raises an incredibly important question. Volunteer bias is indeed a pervasive issue in large-scale cohort studies like the UK Biobank. It's a real headache. The problem is that participants in such studies are often, well, not perfectly representative. They might be healthier, more educated, or from specific socioeconomic backgrounds compared to the general population.
Right. The people who sign up aren't necessarily average Joes.
Exactly. And if you simply perform a GWS on this non-representative sample, your genetic associations might be skewed. They might not reflect the true effect in the broader population. What's truly fascinating here is how researchers have innovated to correct for this. Instead of relying on smaller, potentially less representative surveys to define the general population, they turn to highly representative UK census data.
The official census.
Yes, which boasts an impressive 95% response rate across the entire population. They use this to construct what are called inverse probability, or IP weights.
That's a clever approach. GWAS is a robust source for comparison. Can you walk us through the process? How do you actually create these IP weights? What's the mechanism?
Certainly. The process was meticulously designed, quite clever. First, they restricted the UK census data to include only individuals who would have been eligible to participate in the UK Biobank, specifically those aged 40 to 69 years who lived within the sampling radius of the 22 UK Biobank assessment centers during the recruitment period, 2006 to 2010.
Okay, so comparing like with like.
Precisely, an apples-to-apples comparison. Then, using this meticulously matched census data, they identified and used strong demographic predictors of who actually chose to participate in the UK Biobank versus those who didn't. The most notable and effective predictor turned out to be region of residence, as participation rates varied significantly across different parts of the UK. They then used these factors, region, maybe other demographics, to estimate these IP weights for each UK Biobank participant. Conceptually, an IP weight gives more weight in the analysis to participants who belong to groups that are underrepresented in the study compared to the general population.
And less weight to those overrepresented.
Exactly. It's a statistical rebalancing act. This meticulous approach yielded IP weights for approximately 98% of UK Biobank respondents. These weights then allowed for the construction of the largest weighted genome-wide association study, or GWAS, in individuals of European ancestry to date, encompassing over 376,000 participants. It aims to make the study group more accurately reflect the overall population.
And what were the tangible benefits? Did applying these weights actually change the results? Did they see a difference in the genetic associations?
The benefits are substantial, yeah. They directly address the problem of attenuated or weakened genetic signals that you might get from a biased sample. These IP weights are far more meaningful and precisely estimated because they leverage the immense sample sizes from the census and these directly relevant predictors like region, which effectively capture aspects of the participation bias. Crucially, WGWAS uses these weights to correct genetic associations for that volunteer bias. And what they often find is this results in more predictive genetic effect sizes.
Meaning the estimated impact of a gene looks stronger.
Exactly. The effect size estimate is often further from the null, further from zero or no effect. This is entirely consistent with the idea that volunteer bias can attenuate or dampen the true genetic associations you're looking for.
Can you give a concrete example?
Sure. The UK Biobank, in its initial unweighted form, inherently oversampled individuals with higher levels of education. That's a common finding in volunteer cohorts. After applying the IP weighting, the average years of education within the study population was adjusted downwards, from 13.8 years to a more representative 13.0 years.
Oh, okay.
This demonstrates its effectiveness in making the study population more akin to the general UK population, and thus, hopefully, revealing more accurate genetic effects. It's about getting a clearer, truer signal from your data by correcting for who showed up.
Okay, let's shift gears slightly and talk polygenic risk scores, or PGS. The idea of estimating an individual's genetic predisposition based on many genes sounds incredibly powerful, huge promise for personalized medicine. But where do the challenges lie? Especially when applying them across diverse populations and maybe different socioeconomic contexts, that seems tricky.
The promise of PGS is indeed immense. They work, fundamentally, by summing the effects of thousands or even millions of common genetic variants across the genome to estimate an individual's inherent genetic predisposition to a trait or disease. They hold great potential for revolutionizing personalized medicine. Everything from identifying individuals at high risk who might benefit from early screening to optimizing clinical trial design by selecting participants most likely to respond to a particular therapy. However, a significant, well-documented challenge is that PGS performance varies widely across different contexts.
Contexts like?
Well, demographic factors like age, sex, income, and critically, genetic ancestry. What's fascinating and frankly problematic here is that a PGS developed in one population which, due to historical inequities and funding disparities, has predominantly been European ancestry populations, because that's where the largest data sets were initially built.
Right, the data availability issue.
Yes, exactly.
Yeah.
That PGS often shows substantially reduced accuracy when applied to other populations, like those of African, Asian, or Hispanic ancestry. This is primarily due to fundamental differences in linkage disequilibrium, LD patterns basically. How variants are inherited together on chromosomes differs between populations, and also allele frequencies, how common specific variations are. These genetic differences between populations lead to suboptimal performance of the PGS developed in Europeans, and if we don't address this carefully, it could actually exacerbate existing health disparities.
That's a critical point. So, how are researchers addressing this variability and miscalibration, particularly across different socioeconomic contexts, which, are often overlooked in purely genetic analyses, but are so important for health?
Yeah, this is a major focus now. To tackle this, a sophisticated method called CalPRED has been developed. CalPRED addresses the issue of varying PGS performance by jointly modeling these complex factors to produce truly calibrated prediction intervals.
Calibrated meaning?
Calibrated means that if the model predicts, say, a 10% risk for someone in a specific context, then approximately 10% of individuals in that same context actually do develop the condition. The prediction matches reality for that group.
Okay, got it.
CalPRED's approach involves two key components. First, it models a context-specific mean for each individual. It doesn't assume a single average risk for everyone. Instead, it incorporates how the PGS effect might vary depending on a person's context, like age or income. It even includes PGS context interaction terms to model a varying PGS slope across different contexts. Second, it includes a context-specific variance, which quantifies each context's unique impact on the variation or uncertainty of the prediction interval itself. This allows for more precise and accurate risk estimates tailored to an individual's circumstances.
Can you give us a concrete example of how CalPRED works in practice, particularly regarding those socioeconomic disparities you mentioned?
Certainly. In comprehensive analyses of 72 diverse traits across large biobanks, like all of us and the UK Biobank, CalPRED revealed something quite striking. Traditional PGS prediction intervals sometimes required adjustment by as much as 80% for quantitative traits.
80%, wow.
Yeah, it highlights just how miscalibrated standard predictions can be when context is ignored. For disease traits like type 2 diabetes, T2D, PGS-based predictions were frequently found to be miscalibrated across different socioeconomic contexts, specifically annual household income levels. What they found was that the risk was often underestimated for lower-income individuals and overestimated for higher-income individuals when using standard PGS models.
That's really problematic.
It absolutely is. CalPRED's logistic model, by adeptly incorporating these contexts and their interactions with the PGS, achieves significantly more calibrated predictions across all considered socioeconomic contexts. It demonstrates its profound effectiveness in making these predictions more equitable and accurate. But it's an important caveat. CalPRED requires calibration data that precisely matches the target data's distribution of contexts and their impact on traits. If you're missing context information, you can't properly calibrate. It underscores the need for rich, diverse, and comprehensive data collection, including socioeconomic factors.
Beyond CalPRED, are there other advanced PRS methods being developed specifically to improve performance and predictive yield in non-European populations to tackle that ancestry bias head-on?
Absolutely. The field is rapidly innovating here. It's a huge area of research. Methods like CTSLO and Bridge PRS are really at the forefront. These advanced PRS methods are ingeniously designed to borrow statistical information and insights from larger European GWAs, which often have higher statistical power just due to sample size, while simultaneously optimizing performance for target non-European populations, such as African, Latino, East Asian, and South Asian ancestries.
How do they do that?
Well, CTSLB, for instance, is a model-free approach that combines multiple techniques: clumping and thresholding, which is a common way to select independent genetic variants; superlearning, a machine learning approach for optimally combining multiple prediction algorithms, and empirical Bayes, a statistical method for estimating parameters. It's like a toolkit approach. Breach PRS, on the other hand, is a Bayesian PRS method. It integrates trans-ancestry GWAS summary statistics results from GWAS across different ancestries by retaining all variants within a genetic region, not just the strongest signal. The idea is to better tag potential causal variants that might be shared across ancestries but may be tagged by different SNPs. The core benefit of both these methods is their singular focus on correctly estimating causal effect sizes for prediction and significantly improving predictive yield across a wider range of traits in diverse populations. They're directly addressing those critical health disparity concerns we discussed, allowing us to build truly more generalizable and equitable polygenic risk scores.
Okay, so once we identify a genetic signal, maybe using GWAS or WGWS, the next big challenge is often to pinpoint the exact causal variant, the specific genetic change that's actually driving the association. And doing this across diverse populations, as you've said, adds another layer of complexity. How are researchers getting more precise in this crucial fine mapping step?
This is where multi-ancestry fine mapping methods become absolutely critical. Traditional approaches, especially those combining data from different ancestries, often made an assumption that, well, turned out to be pretty unrealistic.
Which was?
That all causal variants responsible for a trait are shared across all ancestries. In reality, recent research suggests that nearly half of causal SNPs may not be shared across all populations. They might be population-specific.
Ah, okay. That complicates things.
It really does. Assuming they're all shared leads to reduced accuracy. Furthermore, many older fine mapping methods can be computationally very costly, especially with the massive data sets we now have. Or they might oversimplify the problem by assuming only one causal variant per genetic region, which is often not the case for complex traits influenced by multiple variants in the same area.
So how are new methods like MESI addressing these complex challenges, particularly that issue of shared versus ancestry-specific causal variants?
MESIs, which stands for Multi-ancestry Exact Superposition of Single Nucleotide Polymorphism, affects quite a mouthful, is a groundbreaking method. It precisely estimates both shared and ancestry-specific causal signals. It achieves this using a sophisticated statistical model based on a mixture of bivariate normal distributions. This allows it to model the effect of a single genetic variant across two different ancestries simultaneously, capturing both shared and unique effects. This is coupled with a powerful variational inference algorithm, which, conceptually, is a highly efficient computational approach for approximating complex statistical models. It makes the computation scalable even for the massive data sets required today.
And what can it do? Can it really pinpoint specific variants?
Yes, it has remarkable power to pinpoint specific causal signals with high precision. For example, it identified a specific SNP, RS174564, as being causal for triglyceride levels. And importantly, it identified it as shared in both European and African ancestries, and also showed its clear association with the expression of the FADS2 gene,
Which is involved in?
fatty acid metabolism. So finding a variant linked to both triglyceride levels and the expression of a relevant gene across multiple ancestries gives a much stronger clue that it's genuinely causal.
And how does its performance stack up against other methods? Is it really offering a leap forward in precision, getting closer to the true causal variant?
Absolutely. Simulations show MsCAVIAR produces significantly smaller, credible sets. These are the groups of highly likely causal variants within a larger associated region. Basically, it narrows down the list of suspects much more effectively.
Okay.
For example, in simulations, MsCAVIAR had a median credible set size of just 26 SNPs, while an older method like Painter had 77 SNPs, and even a more recent single ancestry method like SUSY had 36 SNPs when applied separately.
So a real improvement in focus.
A real improvement. It demonstrates MsCAVIAR's superior precision in narrowing down the potential causal variants while maintaining comparable statistical power to detect these associations in the first place. It also provides well-calibrated posterior inclusion probabilities, PIPs, for both shared and ancestry-specific signals. This PIP gives researchers a statistical confidence level for each variant being causal. Researchers have already applied this method to multi-ancestry fine mapping on 4 important lipid traits, HDL, LDL, TG, and TC, using data from the UK Biobank for European ancestry and the GLGC Consortium for African ancestry data. The methodology involved meticulous filtering of SNPs and examining functional categories and effect correlations. I should also briefly mention sushi. That's superposition of single nucleotide polymorphism effects in multi-ancestry haplotype context as another advanced multi-ancestry fine mapping method. It's also noted for its high scalability and ability to derive ancestry-specific effect size estimates, which are crucial for downstream applications like building those better multi-ancestry BGS we talked about.
Okay, so we're getting better at finding associations and even pinpointing potential causal variants. But moving from that statistical association to understanding the actual biological mechanism, how the gene works to cause disease, that's the ultimate goal, right? And that's where multi-omics comes in. What does that even mean, and how are researchers using it to achieve deeper biological insight?
That's absolutely right. The ultimate goal is to understand that complete chain of events. How does a genetic variation actually lead to a change in biological function, which then manifests as a disease or trait? Multi-omics is a powerful integrative approach. It combines data from different molecular layers within the same study or the same individuals. We're talking about combining genomics, which is the DNA sequence itself, with transcriptomics, measuring gene expression, how much RNA is being made from a gene,
Right.
proteomics, measuring protein abundance, the actual molecular machines of the cell, and epigenomics, which looks at how DNA is packaged and regulated without changing the underlying sequence, things like methylation. The power lies in seeing how changes at the DNA level cascade through these various layers. It helps uncover the underlying biological mechanisms of disease. It connects the dots.
So it's about seeing the full picture from DNA all the way to disease manifestation. And how do molecular QTLs fit into this intricate puzzle? What are those?
Exactly, the full picture. Molecular quantitative trait loci, or QTLs, are absolutely key pieces of this puzzle. These are specific genetic variants that are associated with quantitative changes in these intermediate molecular phenotypes. For example, eQTLs are genetic variants that influence the expression levels of genes. The E is for expression.
Okay.
pQTLs are variants that influence protein levels, P for protein. And SKUTLs are variants that influence gene splicing patterns for splicing, how the RNA message is put together. These molecular QTLs provide crucial links. They bridge the gap between the initial genetic variation identified in a GWAS and these intermediate molecular phenotypes. They act as critical signposts between your genetic makeup and the ultimate disease outcome. Foundational resources like the GTX study genotype tissue expression have been instrumental in building comprehensive atlases of these genetic regulatory effects across many human tissues. They show us where and how specific genetic variants influence the expression of nearby genes.
So they act like a reference map.
Precisely. A critical reference point for these multi-omics studies. Right. It allows researchers to ask, okay, I found a GWAS hit in this region. Is it also an eQTL for a nearby gene in a relevant tissue? It helps connect the GWAS finding to a potential function.
It sounds like we're drilling down to an incredibly granular level now, far beyond just looking at bulk tissue samples. How are single-cell and spatial genomics contributing to this deeper understanding?
Oh, they are massive advancements offering unparalleled resolution.
Yeah.
The ability to profile gene expression and chromatin accessibility, how tightly packed the DNA is, which affects gene access at a single cell or single nucleus resolution, is revolutionary. It allows researchers to understand how genetic variants act specifically within different cell types within a tissue.
Why is that so important?
Because many diseases, think neurodegeneration involving specific brain cells like microglia, or heart conditions involving cardiomyocytes, primarily affect very specific cell populations. A GWAS hit might only exert its effect in one particular cell type. This level of detail helps pinpoint the exact cellular context of a genetic effect, revealing which cells are most impacted by a genetic variant. It tells you where the action is happening.
And spatial, what does that add?
Spatial transcriptomics, ST, adds another revolutionary layer by preserving the tissue architecture. This means we can not only see what genes are expressed in which cells, but where those cells are located within the tissue.
Ah, so you see the neighborhood.
Exactly. You see the neighborhood. It shows us specific gene expression patterns in their native environment, giving crucial positional context and helping us understand how different cell types interact within the tissue structure. It's like adding a map to the cell data.
And then there's epigenomics, dealing with changes to gene regulation without altering the DNA sequence itself. How are those studies integrated into this multi-omics framework? What are they telling us?
Epigenomic studies are integral, absolutely. They delve into the instructions or modifications that control when and where genes are turned on or off. It's the layer of control on top of the sequence. We're talking about modifications like M6A RNA methylation, which affects RNA stability and translation, and chromatin accessibility, which we measure using techniques like ATAC-seq and ChIP-seq.
Okay.
These methods help us understand how genetic variants might alter gene regulation without changing the underlying DNA sequence. For example, a genetic variant might be in a non-coding region far from any gene. But by altering a regulatory element like an enhancer or promoter, these are like switches or dials for gene expression, it can dramatically change how much protein is made from a gene, or even if it's made at all in a specific cell type or time.
So finding the switches.
Exactly. This is key for pinpointing those critical regulatory elements, which are frequently implicated in disease susceptibility identified by GWAS. And these elements can be located really far from the actual gene they regulate, making them hard to find with traditional methods just looking at the nearest gene. Epigenomics helps map them out.
It's one thing to find all these associations, even potential mechanisms, but another to really prove it, right? What about functional characterization and perturbation studies? How do researchers get direct experimental evidence of causality once they've found a promising genetic lead?
This is where experimental validation comes in, absolutely crucial. Moving beyond just statistical observation to direct evidence of cause and effect. Techniques like CRISPR-based assays are incredibly powerful here. This includes CRISPR interference, or CRISPR, which can reversibly turn down gene expression almost like a dimmer switch, and CRISPR knockout screens, which completely remove a gene's function.
Gene editing tools, basically.
Essentially, yes. They allow researchers to precisely target and perturb gene expression or specific DNA regions to see their direct biological impact. What happens when you tweak this specific thing? For instance, researchers have used CRISPR tiling screens, systematically targeting regions across an area in primary human T cells, specifically T-conv and Treg cells, which are key immune cells. They use this to map out and identify context-restricted enhancers for the CTLA4 gene. CTLA4 is a crucial immune checkpoint, really important in autoimmune diseases and cancer immunotherapy.
Right.
These screens show direct localization of these newly identified enhancers with autoimmunity risk variants found in GWAS. So the risk variant sits right in the functional element. They even went further and used CRISPR to disrupt a specific binding site for a protein called CTCF2 within that region. And they observed a direct biological consequence: increased CD20 expression and decreased CTLA4 expression in those T-conv cells.
Wow, a direct causal link.
A direct causal link between a specific non-coding variant, its regulatory function via that enhancer, and a change in gene expression relevant to disease. And the beauty is, these screens use primary human cells from 2 independent donors to ensure reproducibility, strengthening the validity of their findings. It's powerful stuff. Another crucial technique for direct functional validation is the luciferase reporter assay. It's a very versatile tool used to assess allele-specific effects on gene activity.
How does that work?
Essentially, you take a reporter gene, like luciferase, which produces light, and you put it under the control of a specific DNA sequence containing the genetic variant you're interested in. You test both versions or alleles of the variant. By measuring the light produced, researchers can determine if one version of the variant leads to higher or lower gene activity compared to the other version. For example, in skin pigmentation studies, specific SNPs were inserted into these reporter constructs and tested on gene expression in melanoma cells and primary skin cells, keratinocytes, to see if they directly influenced pigment-related gene activity. And then there's high-C analysis and its higher-resolution cousin, region capture micro-C. These are cutting-edge methods that capture which genomic segments are physically interacting with each other in 3D space within the nucleus.
Because DNA isn't just a straight line, it folds up.
Exactly. It folds into complex 3D structures, and these folds bring distant regulatory elements, like enhancers, into close physical proximity with the genes they regulate, even if they're far apart on the linear sequence. One study, for instance, used this to show significantly greater chromatin interactions, more physical contact between a specific genomic region containing a high blood pressure risk variant, RS1882961, and the promoter regions of 2 genes, NRIP1 and SAMSN1.
But not another nearby gene.
Right, but notably not a third gene, USP25, in the same area. This was specifically observed in vascular smooth muscle cells carrying the high blood pressure allele. This suggests that this genetic variant, likely through its influence on 3D chromatin structure, directly alters the regulation of NRIP1 and SAMSN1, potentially contributing to hypertension. These experimental methods provide crucial direct evidence for the function of genetic variants that statistical associations alone just can't provide.
So we're looking at expression levels, protein levels, DNA folding, all the way from the initial DNA sequence variation. How do we integrate that information with the genetic data from GWAS systematically to really find candidate genes and biological mechanisms at scale?
That's where transcriptome-wide association studies, or TWOs, and proteome-wide association studies, PWAS, come into play. These methods are specifically designed to integrate genetic data directly with measured or, more often, genetically predicted gene and protein expression levels. The aim is to identify the candidate genes and biological mechanisms underlying complex traits and diseases in a more systematic way.
Okay, how does that work generally?
The general process involves first building statistical models, often using data from resources like GTEx or other QTL studies. These models predict gene expression or protein abundance from genetic variants in a given tissue or cell type. You essentially learn which SNPs predict the expression of which genes. Then, these genetically predicted expression levels, or protein levels for PWS, are tested for association with the phenotype of interest from a large GWAS dataset. So TWAS identifies genes whose genetically predicted expression levels are associated with a phenotype. PWS does the same for protein levels. And methods like SUSHI, which we mentioned earlier for fine mapping, also significantly improve the underlying multi-ancestry cis-molecular QTL fine mapping, enabling even more accurate prediction models for use in TWAS and PWAs across diverse populations.
Can you give us a concrete example of their application, showing the power of these integrated approaches? How much extra information do they give?
Sure. In the All of Us Biobank, for example, SUSHI-based prediction models for TWAS demonstrated impressive power. They identified, on average, almost 40 additional associated genes. That's about a 25% increase for 6 white blood cell-related traits compared to existing TWAS approaches.
Wow, that's a significant jump.
It really demonstrates their enhanced capability to uncover novel genetic insights by better modeling expression across ancestries. Furthermore, TWAS, using a specific tool called S-multi-XCAN with summary statistics derived from a GWAS method called MTAG and focused specifically on cardiac tissues, identified 127 genes significantly associated with hypertrophic cardiomyopathy, or HCM. For PWAs, genetically predicted protein levels can be used for nonlinear PWS. This allows for leveraging much larger sample sizes than studies using directly measured proteins, and it helps isolate the genetic component from environmental confounders that also affect protein levels.
Right, protein levels can fluctuate a lot.
Exactly. So using the genetically predicted component helps to zero in on the potential causal role of genetic variation acting through protein abundance.
And what about conditional transcriptome-wide association studies, or CTWS? How does that go a step further? Does it untangle even more complex relationships?
CTWS is an advanced gene-based association method that moves beyond simple correlations between predicted expression and the trait. It simultaneously models the effects of both the genetic variance in a region and the imputed gene expression on a specific trait, all in one model.
Okay, what's the advantage of that?
Its key advantage lies in its ability to account for pleiotropic effects, where a single variant might affect multiple genes or the trait directly, and also linkage disequilibrium, LD, more robustly than prior methods. It tries to tease apart whether the association is truly mediated by the gene's expression or if the gene's expression is just correlated with the trait due to LD with the actual causal variant.
So it helps distinguish correlation from causation a bit better.
Precisely. It can even be viewed conceptually as a two-stage Mendelian randomization method, which, as we'll discuss later, is a powerful approach for inferring causality. This provides a more robust framework for detecting truly causal genes, as opposed to genes that are merely correlated or in LD with a causal gene nearby. This precision can directly reveal disease mechanisms and highlight more promising therapeutic targets. It also has the added benefit of being able to estimate the proportion of trait heritability attributable specifically to gene expression, giving us a clearer picture of how much of a trait's genetic basis is mediated through gene regulation.
With so many associations being uncovered by GWAS, TWAS, all these methods, it must be like trying to find a needle in a haystack sometimes to identify the true effector gene, the one actually causing the disease signal, especially when, as you said, it's often not the nearest gene in the genome, which is a misleading assumption people used to make. How do researchers navigate this complexity? How do they prioritize?
That's a critical challenge, absolutely. And it's one of the biggest bottlenecks in moving from GWAS discoveries to actual therapeutic development. Pinpointing the true effector gene from a broad GWAS signal, often containing many genes in LD, is incredibly difficult. And yes, most GWAS associations are found outside of protein coding regions in regulatory DNA. So simply picking the nearest gene to a GWAS hit is often wrong. It's a flawed heuristic.
Right.
This is where sophisticated integrative frameworks like FLAMES come into play. These frameworks aim to systematically identify the most likely effector gene at GWAS loci, the specific gene, whose variation actually mediates the observed association with disease. They help us cut through the noise in the LD to find the true biological culprit.
And how does FLAMES achieve this? What makes it potentially more effective at sifting through all that data to find the causal link?
FLAMES uses a really ingenious two-pronged approach. It combines diverse types of evidence into a single, well-calibrated prediction score for each gene in a GWAS locus. First, it integrates SNP to gene evidence. This directly links a genetic variant, SNP, implicated by GWAS or fine mapping to a potential target gene. This includes data from chromatin interactions like Hi-C, showing physical contact, various QTLs, EQTLs, PQTLs, QTLs linking SNPs to gene expression, protein levels, or splicing, and even simple genomic proximity, though this is weighted less heavily because it's less reliable.
Okay, direct links. What's the second prong?
The second prong incorporates convergence-based evidence. This looks at a broader systems-level view. It tries to identify genes that are consistently enriched across entire GWAS for involvement in shared biological pathways or functions related to the disease.
So looking for patterns across many GWAS hits.
Exactly. It's looking for patterns that suggest a gene is involved in the overall disease biology, not just associated with a single GWAS hit. It asks, does this gene keep popping up in relevant contexts? By training on unbiased data, such as results from exome-wide association studies, XWAS, which look directly at coding variants and are less prone to LD confounding for linking to the right gene, FLAMES aims for higher generalizability. And it has been shown to significantly outperform approaches that rely on only one type of evidence, like just using EQTLs or just proximity. This truly helps bridge the gap from a mere statistical association to understanding biological function and ultimately accelerates the path to therapeutic development by giving researchers high-confidence effector gene candidates to investigate further.
Okay, let's talk about causality directly. Establishing causality in observational studies is notoriously tricky. We often say correlation does not equal causation. But genomics seems to offer a powerful way to get closer to cause and effect. How does Mendelian randomization, or MR, help us here? What makes it so potentially robust?
Mendelian randomization, or MR, is an incredibly powerful and increasingly popular method. It cleverly leverages the fundamental principles of genetics to infer causal relationships between variables. It uses genetic variants as instrumental variables, essentially, naturally occurring random assignments to infer causal relationships between modifiable exposures, like lifestyle factors such as BMI, smoking, or even blood lipid levels, and health outcomes, like heart disease risk.
How does the random assignment part work?
Because the genetic variants you inherit are largely determined randomly at conception, based on which alleles you get from each parent, they act like a natural experiment. Your genes are assigned before environmental factors or lifestyle choices come into play. The core principle, particularly with the most common method, inverse variance weighted, or IVWMR, relies on a crucial assumption that the genetic variants used as instruments for the exposure, say variants robustly associated with BMI, have no direct causal effect on the outcome, heart disease, except through their effect on the exposure, BMI.
So the gene only affects the outcome via the exposure pathway.
Exactly. Because these genetic variants are randomly assigned at conception, they are generally not confounded by the environmental or lifestyle factors that plague traditional observational studies. This allows for much stronger causal inferences.
That's a great explanation of the core idea, but what if that key assumption that the gene only works through the exposure doesn't hold? What if the gene does other things too? How do researchers test for that?
That's where sensitivity analyses come in, and they are absolutely crucial for validating MR findings and making sure you're not being misled. You can't just run one MR method and call it a day. Methods like MR-Egger regression, weighted median MR, and others are specifically designed to test for and potentially adjust for horizontal pleiotropy.
Horizontal pleiotropy.
That's the term for when a genetic instrument influences the outcome through pathways other than the exposure of interest. It violates that core assumption. If significant pleiotropy is detected and isn't accounted for, it can invalidate the causal inference. So these sensitivity analyses help researchers understand the robustness of their findings. Do different methods give consistent results? Is there evidence of pleiotropy? It helps determine if the causal link truly holds up to scrutiny or if there might be alternative explanations.
Can you give us some diverse, real-world examples of MR in action, showing its versatility in teasing out these complex causal relationships?
Certainly. MR analyses have been widely applied to explore a huge range of biological and clinical questions. For instance, researchers have used MR to test potential causal associations between sarcoidosis risk and 6 modifiable risk factors, things like BMI, waist-hip ratio, smoking initiation, alcohol intake, coffee consumption, and physical activity using large GWAS meta-analysis data. This helps prioritize which factors might be causal targets for prevention. In another compelling example, MR was used to validate causal associations between genetically predicted platelet count, PCT, and monocyte count, MONO, types of blood cells, and the presence of JAK2V617F clones. This specific mutation is a key driver in certain blood cancers called myeloproliferative neoplasms. The study demonstrated that higher genetically predicted platelet and monocyte counts were causally linked to having these mutant clones, and the causal estimates were more accurate when using larger cohorts like the UK Biobank WES data. This gives a clearer picture of the early pathogenesis of these disorders.
Interesting. Other areas?
Oh yes, MR has been employed in 2 sample studies, using summary data from separate GWS to test the causal association of heart muscle contractility, left ventricular contractility, with hypertrophic cardiomyopathy, HTM, including both non-obstructive and obstructive forms. And beyond that, it's being used constantly to explore intricate causal relationships among factors like the gut microbiome composition, various blood lipid levels, inflammatory markers, and complex conditions like heart failure, Alzheimer's disease, depression, you name it. It's a really versatile tool for trying to establish causality where traditional observational studies struggle with confounding.
Clinical trials, RCTs, are often called the gold standard for proving causality and the effectiveness of new treatments. But as we know, they're incredibly expensive, time-consuming, and sometimes the people in them don't fully reflect real-world patient populations. Is there a way to sort of emulate these trials using the vast data we now have in biobanks? And how does genetics play a role in making that emulation more powerful?
You've hit on a critical point and a really exciting area of development. RCTs are indeed the gold standard for causal inference in medicine, but they absolutely come with significant costs, time commitments, and often recruit highly selected populations, which might limit how generalizable the findings are to everyday patients. This is precisely why target trial emulation in biobanks is gaining so much traction. It's a framework that uses the rich, longitudinal observational data from large biobanks like UK Biobank or FinnGen, often combined with genetic insights, to statistically mimic or emulate a hypothetical randomized controlled trial.
How does that work? How do you mimic a trial?
The idea is to very carefully design your observational analysis to answer the exact same question that a hypothetical ideal RCT would answer, even if actually conducting such an RCT isn't feasible due to cost, ethics, or time. You explicitly define the target trial's protocol eligibility criteria, treatment strategies, outcome definition, follow-up period, analysis plan, and then apply that protocol to the observational data.
Okay, so how does genetics specifically bolster this emulation, making it more robust and reliable than just a very careful observational study?
Genetics adds a powerful layer, particularly for strengthening causal inference. By leveraging genetic variants as instrumental variables, much like in Mendelian randomization, researchers can significantly strengthen causal inference regarding treatment effects or exposure outcomes in these real-world settings. It helps control for unmeasured confounding.
Right, the confounding issue again.
Exactly. Traditional observational studies are always plagued by potential confounding, where unmeasured factors influence both the exposure, like starting a medication, and the outcome, like having a heart attack, creating a spurious association. Incorporating polygenic risk scores, for example, within the trial emulation framework can actively help detect and mitigate these confounding biases. A PGS can serve as a proxy for baseline risk, helping to adjust for differences between groups. This provides far more robust causal estimates than traditional observational studies could offer alone. Furthermore, this approach explores the valuable role of using polygenic risk scores for prognostic enrichment and predictive enrichment within these emulated trials.
Prognostic and predictive enrichment, can you clarify those?
Sure. Prognostic enrichment means using a PGS to identify individuals who are at higher baseline risk of developing the disease outcome you're studying. Focusing a trial, real or emulated, on these high-risk individuals can make it much more efficient, requiring fewer participants to see an effect. Predictive enrichment means using a PGS, perhaps interacting with other factors, to identify individuals who are most likely to respond positively or negatively to a particular intervention being studied.
Identifying responders.
Exactly. This is highly relevant for stratified medicine and precision healthcare, where we aim to tailor treatments. It helps identify who is most likely to benefit from a specific intervention, making future clinical trials, whether real or emulated, far more efficient and targeted. It's essentially using the rich observational and genetic data we already have to get closer to the certainty of an RCT, faster and cheaper.
With data exploding at just an unprecedented rate in genomics, machine learning isn't just a buzzword anymore. It seems like it's becoming indispensable in virtually every field, including this one. How is ML transforming how we conduct and interpret GWAS, maybe moving beyond some of the traditional statistical methods?
You're absolutely right. Machine learning techniques are indeed being integrated across virtually all stages of genomic studies now. It's not just hype. It's becoming fundamental. They're touching everything from sophisticated data imputation, filling in missing data points and handling missing values, to highly accurate risk prediction models, and even teasing apart very fine-scale population stratification or structure. They're becoming essential tools for extracting meaningful insights from the incredibly complex and high-dimensional genomic data sets that we now routinely generate. Think of it as empowering researchers to find patterns and make predictions that traditional statistical methods might struggle with or miss entirely in the sheer volume and complexity of the data.
Can you give us an example of how ML is specifically assisting GWAS studies to overcome traditional limitations? Like that challenge you mentioned earlier of obtaining high-quality phenotypic data for everyone.
Yeah, a key innovation here is ML-assisted GWAS, and a particularly interesting statistical framework called POP-GWAS. Traditionally, as we discussed, GWAS power can be limited by the availability of high-quality phenotypic data, precise measurements of traits or disease states. This data can be expensive, time-consuming, or invasive to collect, like requiring imaging or biopsies, which ultimately reduces the sample size with full data, limiting statistical power for detecting genetic associations.
Right.
ML-assisted GWAS aims to overcome this by leveraging machine learning algorithms to infer or predict phenotypes for samples where the measurement is missing, using other available data like demographics, basic lab tests, or even genetic data itself. Or it can be used to combine data from both labeled phenotype known and unlabeled phenotype unknown datasets.
But doesn't predicting phenotypes introduce errors? Could that lead to false discoveries?
That's exactly the concern that earlier ML-assisted approaches sometimes faced. They showed promise, but carried risks of false positive associations if not handled carefully. POC-GWAS specifically addresses these limitations. It's a statistical framework that redesigns the GWAS analysis specifically to work with these ML-imputed outcomes in a statistically rigorous way. It's considered an assumption-free and no-harm method. What this means conceptually is that it makes no strong assumptions about the predictor variables used for imputation, the specific ML algorithm chosen, it could be a random forest, neural network, anything, or even the quality of the phenotype imputation itself. Crucially, it's mathematically proven to enhance statistical power by leveraging the imputed data, while rigorously ensuring the validity of the identified associations controlling the false positive rate properly.
Okay, that sounds powerful. Has it been used?
Yes. For example, it's been successfully applied to bone mineral density, a complex trait derived from dual-energy X-ray absorptiometry, DXA imaging, which isn't available for everyone in large cohorts. POP-GWAS, using imputed bone density, not only identified novel genetic loci associated with bone density, but also revealed skeletal site-specific genetic architecture, finding variants specific to, say, hip versus spine density. It showcased its ability to find new insights where traditional GWAS might have struggled due to limited direct phenotypic data.
That's incredibly powerful, using ML to effectively create more phenotypic data in a statistically sound way. What other specific ML applications are being deployed to make sense of these vast biobank datasets?
Oh, there are many, each tackling a specific challenge. Autocomplete, for instance, is an ML method specifically designed for highly accurate data imputation, filling in missing genetic or phenotypic data points in extremely large datasets like the UK Biobank. Studies showed it outperformed other established imputation methods like MsForest and MIE, especially when there's a higher percentage of missing data. Apparently, a technique called copy masking was important for its accuracy. The random forest classifier is another versatile machine learning tool used in various ways. It was used in the UK Biobank analyses to recognize potentially low-confidence calls for tandem repeat expansions, or TREs, based on various characteristics of the genomic locus. TREs are these tricky, repetitive regions sometimes linked to neurological disorders. Random forests were also applied in fine-scale genetic structure analysis of Japanese populations. They used it to classify individuals into distinct genetic clusters based on geographical origin and other data, revealing nuanced population substructures that are really important to account for in genetic association studies within Japan. Then there's Survival FM. This is a sophisticated machine learning model specifically designed to enhance cardiovascular risk prediction. It goes beyond standard models like Cox regression. It does this by comprehensively modeling complex interaction terms among a wide array of standard clinical risk factors, like those used in the QRSK3 score. Standard models often only consider additive effects, but Survival FM looks at how risk factors might amplify each other, and it showed significant improvement. Better discrimination ability to distinguish high-risk from low-risk individuals, and better reclassification correctly moving people into more accurate risk categories. It captured about 6.7% more cardiovascular events compared to a standard Cox model at the clinically relevant 10% risk threshold, while crucially maintaining proper calibration, meaning its predicted risks accurately match the observed risks in the population.
And what about just the sheer computational challenge? As biobanks grow to millions of samples, running a GWAS itself becomes a massive task. Are there new ML-inspired tools tackling efficiency without sacrificing power?
You're right. The trade-off between computational efficiency and statistical power is a constant battle as data sets explode in size. Novel computational GWAS tools like QuickDraws are addressing this head-on. QuickDraws aims to significantly increase association power for both quantitative, like height, and binary, like disease status traits. It does this by leveraging a powerful statistical approach called a spike and slab trier on variant effects. Conceptually, this helps the model focus on identifying true genetic signals while effectively shrinking noise or null effects towards zero. It combines this with stochastic variational inference for efficient computation and, crucially, GPU acceleration. Using graphics processing units allows for massive parallel computation, dramatically speeding up the complex calculations involved.
So faster and maybe even more powerful.
Yeah, exactly.
QuickDraws allows performing large-scale GWAS incredibly efficiently without sacrificing power. In fact, simulations and real data applications showed it often had superior power identifying a higher percentage of true associations compared to widely used methods like Regini and FastGWA, all while maintaining comparable or even better computational costs. It's a significant leap towards maximizing discoveries from these massive genetic datasets without getting bogged down for months in computation time.
And beyond looking at individual variants or traits, how are researchers using ML or advanced computation to look at bigger pictures, like how different diseases cluster together or progress over time in populations? This is where methods like 3D disease network analysis come in. It's a highly refined approach aimed at identifying robust clusters of diseases and tracing key progression pathways after an initial or index disease diagnoses like maybe depression or diabetes. It integrates both temporal information, the order and timing of diagnoses, and non-temporal associations, diseases that just co-occur, using sophisticated statistical techniques like regularized partial correlations within a three-dimensional framework: patient, disease, time.
Why is that needed? What are the limitations of older network methods?
Existing network analysis techniques often struggle with statistical power due to the huge number of potential disease pairs, multiple testing burden. They can also find spurious associations and often rely on only one type of data, like just co-occurrence ignoring timing, which makes it hard to validate findings across different data sets or healthcare systems. This newer 3D approach aims to overcome those limitations. The goal is to produce more clinically interpretable disease clusters and progression patterns that show high consistency across large, independent population-based cohorts, like validating findings from Swedish national registers in the UK Biobank. This provides a more nuanced understanding of disease multimorbidity, how diseases cluster together, how they progress over time, and what might be the causal pathways linking them, which can directly inform potential clinical interventions or preventative strategies by identifying high-risk trajectories.
That's a truly comprehensive suite of computational and ML tools. Beyond these, how are researchers employing maybe even more advanced statistical modeling techniques? And how are they actively considering gene-environment interactions to deepen our understanding of genetics beyond just simple associations?
Yeah, the field is definitely pushing beyond standard GWAR models to capture more nuanced aspects of genetic influence. For instance, family-based GWARs or FGWAS and the estimation of direct genetic effects, DGEs, tackle a really significant challenge in traditional population-based GWAS, confounding biases.
We talked about volunteer bias, but what other confounding is there?
Well, traditional GWAS can be confounded by population stratification, subtle genetic differences between subgroups in your study that happen to correlate with disease prevalence, making it look like a genetic association when it's really just ancestry,
Mm-hmm.
or by indirect genetic effects.
Indirect effects, like what?
Things like genetic nurture, where parental genotypes influence an offspring's phenotype, not directly through inherited genes, but through the environment the parents provide, which is itself influenced by the parents' genes. Or assortative mating, where individuals with similar traits, and thus often similar genotypes for those traits, tend to mate, which can inflate genetic associations.
Okay, so how does FGWA get around that?
FGWAS addresses this by incorporating parental genotypes directly into the GWAS regression model, using data from trios, parents and offspring, or larger families. By exploiting the random segregation of alleles during meiosis, the process of forming sperm and egg cells, which is like a natural randomization event within families, FGWAS can robustly estimate the direct genetic effects, DGEs, of the offspring's own genotype, separate from these indirect effects.
So it isolates the effect of the genes the child actually inherited.
Exactly. It inherently removes or adjusts for confounding biases from population structure and these indirect genetic effects. This provides estimates of heritability and individual SNP effects that are theoretically free from these confounds, offering a much clearer dissection of direct genetic influences from indirect or environmental ones. It's been effectively used, for example, to estimate less confounded genetic correlations for complex traits like height and educational attainment.
That sounds like it provides a much cleaner genetic signal. What about traits that change over time? Standard GWAS usually uses just a single measurement, but many conditions, like obesity or blood pressure, evolve throughout a person's life.
That's another key limitation of standard GWAS. For many complex traits that change over a lifespan, and where genetic effects might not be constant, maybe they only kick in during puberty or later in life. Traditional GWAS using single measurements falls short. Longitudinal data analysis for repeated measures is crucial here. Statistical methods, often based on linear mixed models, LMM, are employed to utilize repeated phenotype measures collected over time from the same individuals within large cohorts. This approach allows for the detection of genetic variants whose effects actually vary over time. It provides a much more dynamic understanding of genetic influences on traits like, say, childhood BMI, which changes dramatically from birth through adolescence. This can help identify genetic time bombs or accelerators whose effects only become apparent at certain life stages, which a single snapshot GWAS would miss.
And are we still looking for rare variants? We hear a lot about common variants from GWAS, but rare variants can sometimes have a strong impact, right? How do we find those?
Absolutely. While common variants identified by GWAS explain a substantial portion of complex trait heritability, rare and low-frequency variants are increasingly recognized for their significant role in disease, especially in severe or early-onset conditions, or sometimes even common diseases. But traditional single-variant GWAS testing one SNP at a time has very limited statistical power for these rare variants simply because they occur so infrequently in the population. You might only see a few copies even in a large study.
So how do you boost the signal?
Rare variant aggregation testing is the main strategy. Instead of testing each rare variant individually, these methods aggregate the effects of multiple rare genetic variants found within a specific gene or a predefined genomic region, like an enhancer. You then test the collective effect of all rare variants in that unit for association with the trait. This approach generally assumes that multiple different rare variants within the same functional unit might disrupt its function similarly and collectively contribute to the trait, thereby increasing statistical power compared to testing each one alone. It's crucial for uncovering the full genetic architecture of complex traits and has been successfully applied, for instance, to discover novel genes associated with metabolite levels in the blood, where rare coding variants often have large effects.
And what about in silico modeling? That sounds futuristic. Can we use computational models to simulate biological processes to understand gene function and maybe even identify new biomarkers without doing wet lab experiments?
Yes, in silico, or computational modeling, is becoming incredibly powerful, acting almost like a virtual laboratory to complement experimental work. Personalized whole body models, or WBMs, are a fascinating example. These are complex computational models of human metabolism that can be personalized with an individual's unique data, such as their gut microbiome composition measured from stool samples. Studies have shown these personalized models can accurately capture observed metabolic changes in individuals.
Okay, so you build a virtual human metabolism. Then what?
Then you can use it to study gene function and identify potential biomarkers through computational simulation. As an illustration, researchers generated hundreds, 569, of these microbiome-personalized WBMs. They then used them to calculate the effect size of an in silico meeting, simulated in the computer knockout of the KYNU gene on metabolite excretion into urine.
KYNU encodes an enzyme, right?
Yes, kynureninase, involved in tryptophan metabolism. This in silico approach successfully replicated known biomarkers of kynureninase deficiency, things seen in patients with mutations in this gene. And excitingly, it also identified a potentially novel biomarker, 8-methoxykynurenate, whose prediction was consistent with findings from real-world population studies and known inherited metabolic disorders. It allows for rapid, large-scale hypothesis testing about gene function and metabolic consequences that would be incredibly slow or expensive to do experimentally.
That's a really unique way to bridge genetics and metabolism. What about environmental factors? We know genes don't act in a vacuum. How are environmental exposures being integrated more actively into genetic mapping, moving beyond just saying nature versus nurture?
Environmental perturbation, combined with genetic mapping, is a truly innovative strategy that actively seeks to uncover gene-environment interactions, or GXE. It aims to find genetic variants whose effects on a trait are modified by a specific environmental exposure.
How do they do that experimentally?
It involves experimentally manipulating biological samples, for example, taking human blood cells from many different donors in vitro in the lab, and exposing them to specific drugs, chemicals, environmental toxins, or even immune stimuli. Then you perform GWAS on the resulting cellular responses. These responses are often measured using high-throughput techniques like flow cytometry, which can measure dozens of cellular features simultaneously. This approach is partly inspired by successful studies in model organisms like yeast or worms, where environmental stimulant can be precisely controlled to reveal GXE interactions that might otherwise remain hidden or require astronomically large sample sizes to discover in humans under normal, uncontrolled conditions.
So you stress the cells and see who reacts differently based on genetics.
Exactly. By characterizing these cellular responses to specific perturbations, these perturbed GWAS can identify genetic loci associated with specific non-resting or stressed cell states. This provides a powerful blueprint for discovering new disease genes and mechanisms that are relevant specifically to how the body reacts to various external factors. It reveals these gene-environment quantitative trait loci, GXEQTLs, that would likely be missed in standard GWASs looking only at baseline states.
Finally, how are these advanced methods addressing challenges in areas like imaging genetics, studying traits like brain structure or facial features, where the phenotype, the measurable characteristic, is itself very complex and high-dimensional?
Yeah, for complex morphological traits, like subtle variations in facial features captured by 3D cameras, or differences in brain structure measured by MRI, genetic studies often face significant challenges. These include potentially limited sample sizes, because collecting high-resolution imaging data on huge numbers of people is expensive and logistically difficult, and also the sheer complexity of the phenotyping itself. How do you accurately quantify a face shape or brain structure?
Right.
Combined GWAS, or CGWAS, is a multivariate statistical method specifically designed to increase statistical power in these scenarios. It works by effectively combining the results from multiple single-trait GWAS performed on correlated traits derived from the complex phenotype. It often leverages advanced phenotyping methods first, like using machine learning for 3D facial landmarking or segmenting brain regions, to break down the complex image into many simpler, correlated quantitative traits.
Okay, so analyze many simple features together.
Exactly. Crucially, CGWAS accounts for the intricate correlations among these derived traits, which are often influenced by shared genetic and non-genetic factors. By analyzing them jointly, it boosts power. This leads to increased statistical power and the discovery of novel genetic loci that might be missed by standard single-trait GWAS, which analyze each derived feature in isolation. This method is particularly valuable when dramatically increasing the sample size isn't feasible, allowing researchers to extract a maximum genetic insight from existing, rich imaging datasets.
Ultimately, all this incredible science, all these complex methodologies, they aim to improve human health, right? So how are these advanced methods actually moving us closer to actionable clinical insights? How do we bridge that gap from research discovery to direct patient benefit?
The translation of these profound discoveries into tangible clinical benefits is indeed the ultimate goal, and it's a major focus. A crucial aspect here is accurately interpreting the clinical significance of the genetic variants we find, especially the vast number of variants of uncertain significance, or VUS.
Right, the ones where we don't know if they're harmful or benign.
Exactly. These are variants whose impact on health isn't yet clear, which creates huge challenges for clinical reporting and genetic counseling. There's a recognized need for more granular data, particularly regarding disease penetrance.
Penetrance meaning?
The probability of actually developing a disease, given that you carry a specific genetic variant, it's often not 100%. Traditional classifications are often binary, pathogenic, or benign, but we really need to move towards more quantitative risk estimates. What is the actual risk percentage associated with this variant, perhaps at different ages? Solutions involve integrating real-world population data from large biobanks. How common is the variant? Is it seen in healthy older people? Age-stratified information, because penetrance of some variants changes dramatically with age, and leveraging advanced statistical frameworks, like Bayesian methods, to refine these penetrance estimates. The goal is to move from those qualitative black-and-white interpretations to nuanced quantitative risk estimates that are truly clinically actionable for patients and their doctors.
And how is all this data and methodology helping to optimize clinical trials? Those are so vital for actually bringing new treatments to patients.
Genetic data is proving invaluable in optimizing clinical trial design, making them smarter, more efficient, and more likely to succeed. As we discussed earlier with trial emulation, using large observational biobank data combined with genetics can help researchers assess potential interventions and detect confounding in real-world settings, providing early insights into potential drug effects or side effects even before launching a large, expensive Phase 3 trial.
And the enrichment strategies.
Right. Prognostic enrichment strategies, which use polygenic risk scores to identify individuals at higher baseline risk of an outcome, or a specific disease progression, can significantly reduce the sample size needed for randomized controlled trials. If you focus on people more likely to have the event, you need fewer participants overall to see if a drug prevents it.
Makes sense.
This makes trials much more efficient and cost-effective, potentially allowing new therapies to reach patients faster. And Mendelian randomization, too, plays a role by helping to identify likely confounders and establish more robust causal relationships between genetic variants, biomarkers targeted by drugs, and disease outcomes, further refining trial design by ensuring we're testing the right drug mechanisms in the right populations.
And going back to those effector genes, you mentioned the ones truly driving the disease signal at a GWAS locus. How important is prioritizing them for actually developing new therapies? It sounds like identifying the right target is half the battle.
Identifying the actual effector genes responsible for a disease signal from the thousands of potential genetic associations is a major ongoing task, and yes, it's absolutely where much of the field's focus is now directed for translation. This involves that systematic integration of various functional genomic data. We talked about eQTLs, pQTLs, chromatin interactions, epigenomics, and utilizing advanced computational tools like FLAMES or similar frameworks to predict the most likely causal genes. There's a strong and growing call within the research community for standardized reporting and open access catalogs of these predicted effector gene lists.
Why is that so important?
It's absolutely vital to ensure their interpretability, transparency, and reusability by the broader scientific community, including researchers in pharma and biotech. When researchers can easily access, compare, and potentially validate these high-confidence effector gene candidates from multiple studies and using multiple lines of evidence, it directly accelerates the path from basic genetic discovery to the development of novel therapies targeting those specific genes. It brings us closer to truly targeted, mechanism-based treatments.
With all this power, all this potential to understand and maybe even predict health outcomes, there are obviously critical responsibilities. What are the ethical, legal, and social implications, the ELSI issues, that are shaping the future of genomic research?
This is an incredibly important area and one that is absolutely central to responsible genomic science. You cannot do this work without constantly considering the ethical dimensions. Genomic research, especially when conducted at population scale and across diverse ancestries, raises significant ethical considerations that must be carefully navigated. These include ensuring benefit sharing with the communities whose data contributes to these discoveries. How do we make sure the benefits of the research, like new diagnostics or treatments, actually flow back to all contributing populations and not just become concentrated in wealthier nations or groups? It also involves rigorously respecting informed consent, ensuring participants truly understand how their data will be used now and potentially in the future, and data sovereignty, particularly for indigenous populations or specific communities who may want more control over their collective genetic information.
And privacy and discrimination.
Absolutely. Protecting participant privacy from misuse is paramount, especially as datasets become larger and potentially re-identifiable. and diligently addressing potential issues of discrimination, stigmatization, and the reinforcement of harmful racial or ethnic stereotypes based on genetic findings is an ongoing challenge that requires constant vigilance and proactive measures in how we conduct and communicate the research.
So it's not just about the science itself, but also the societal impact and how we talk about it responsibly.
Absolutely. Guidelines from prominent bodies like the U.S. National Academies of Sciences, Engineering, and Medicine strongly emphasize the careful and nuanced use of population descriptors in genetic research, moving away from outdated and socially loaded racial categories towards more precise descriptions of genetic ancestry while acknowledging the impact of social determinants of health. The goal is to avoid misconceptions and promote inclusivity. Furthermore, effective risk communication is crucial. How do we explain complex, often probabilistic genetic risks to individuals in a way that is understandable, culturally sensitive, and doesn't cause undue anxiety or fatalism? This is especially important for the responsible and equitable clinical implementation of things like polygenic risk scores. The scientific rigor must always be matched by ethical responsibility, ensuring that the incredible power of genomics benefits all of humanity equitably.
So, wrapping up, what does this all mean for you, the listener, thinking about the future of genomic research? We've seen that the field is moving in truly exciting directions, evolving at just breakneck speed.
Yeah, if we try to connect this all back to the bigger picture, several core trends are really defining what looks like a much more integrated and hopefully inclusive future for GGOAs and genetic discovery. First, there's the undeniable importance of massive scale. The increasing reliance on these vast biobanks and global consortia is absolutely enabling the detection of increasingly subtle genetic signals relevant to common, complex diseases. Size matters here. Second, we have multidimensional insight. Integrating these diverse data types, DNA, RNA, protein, cellular phenotypes, epigenetics, 3D structures, is crucial for moving beyond just finding statistical associations to uncovering the actual causal biological mechanisms of disease. We need the whole picture. Third, there's the rise of computational sophistication. Advanced statistical methods and machine learning aren't just optional tools anymore. They're essential partners for extracting meaningful insights from these complex, high-dimensional datasets, and importantly, for proactively addressing inherent biases like population stratification, volunteer bias, and confounding. Fourth, the entire field is relentlessly driven by clinical relevance and translation. The ultimate goal is always better diagnostics, more accurate prognostics, and more effective therapies. This requires robust variant interpretation, optimizing clinical trials using genetics, and systematically identifying those crucial effector genes.
And finally,
And finally, perhaps most importantly, there's a paramount and growing emphasis on inclusivity and responsibility. There's a critical recognition of the need for greater diversity in genomic studies, diversity in genetic ancestry, socioeconomic backgrounds, geographic locations to ensure equitable benefits across all populations. And this absolutely requires careful consideration of all those ethical implications and transparent, culturally sensitive communication.
so for you, whether you're deep in research yourself, maybe trying to translate these discoveries into clinical practice, or just incredibly curious about the cutting edge of human biology, it's clearly a time of immense opportunity and rapid change. Which leads me to a final thought to leave you with. If these powerful methodologies are uncovering so much about human health and disease, what new kinds of collaborations, maybe new data sharing frameworks, will be absolutely essential to truly realize their promise for global health equity, especially given the ongoing, very real challenges of data privacy and ensuring that the benefits derived from genomic data actually reach all the diverse populations who contribute to it? That's something worth pondering.